Okay, so here we are in the cardiovascular portion of our assignment here, working on the heart. So this is going to go into the heart playlist. This is our lab. I've drawn the lab here. This is a turtle whose head has been pulled up and out. He was trying to hide from us, but we wouldn't let him. This is the ventral side up. Then we have a 2x4 here. We had a hammer and we had a nail, large nail, and it was my job to pith or to kill the turtle by placing the nail at appropriate location and pounding it through the head of the turtle, while my partner Stephanie, using the pliers, held the turtle's head onto the 2x4. My partner was my best friend, Stephanie, on her way to physical therapy school in Portland, Oregon, and she had been a Vipassana Buddhist practitioner, vegan. We had to kill an animal every single week in Dr. Close's physiology class in the year of our Lord 1982, I think. At any rate, I took the role of protecting her from ruining her karma by killing all the animals myself. So in this particular case, we had the turtle. Once the turtle was pithed and was presumably brain dead, but the heart was still beating, we had a hacksaw and we cut away the shell around the front of the animal, and then we exposed the heart, and we found the vagus. Okay, and the vagus is hooked up to the heart as indicated in your chapter, same as a human being. And the purpose of the vagus actually is to slow the heart sufficiently so that the end diastolic volume fills and we get a heartbeat. The heartbeat is continuous and long, along diastole, so that the heart can put out a proper cardiac output. So it actually has two ways of depolarizing, first with sodium and then with calcium. So we get the plateau with the calcium. The idea here is that we were going to measure using this armature hooked up to a stand next to our little setup with the turtle there, and the armature would end up with a pen positioned against graph paper on a slowly rotating cylindrical chirometer, which I've drawn here in the direction of the rotation, and as it rotates, you can see the pen is drawing the heartbeat. Right here is the pen. So this would have been graph paper. I decided not to draw in all of the cross hatches of the graph paper, but you get the idea. So the idea here is that it's a grisly procedure, I'll tell you this. But the first thing that we did was when we exposed the heart, we bathed it in epinephrine, and the heart rate just took right off. Okay, and then we rinsed the heart with sterile saline, and that cleared the epinephrine, and the heart went back to its resting heart rate. And then rather than save the turtle, which was, I think, at that point beyond saving, to be perfectly honest, since we piffed the brain and we took away his shell, then we could do the final procedure, which is to cut the vagus. Once you cut the vagus can no longer slow down the heart. So even in the absence of epinephrine, the heart sped up to about whatever it was, the equivalent of a human's 110, whatever the turtle version of that would be. Okay, so turtles are not highly aerobic individuals, but they nevertheless have a heart which proceeds to that level. I wanted to present this to you so that you didn't have to go through the, how shall I say, the grisly experience of killing an animal just for every 2 or 4 people, however many, in order to prove what is so easily just described here in a diagram. In medical school, we had a dog, which was, they were kept in dog kennels for the very purpose of this experiment, in which the dog went into a harness for open heart surgery. And we didn't pith the dog. We gave it very heavy-duty anesthesia, but the heart was still functioning. And I pled with the head of the lab professor, that shouldn't we, as medical students, we were second-year medical students on our way to our MD, and I tried to convince him that, we were intended to save lives, not kill lives, and that maybe we should, rather than cut, we sort of got the idea of what was going on, and I didn't think we should cut the vagus, but he insisted that the dogs had been raised and bought for that purpose, and that they would be interred, and that there was a whole system set up that would not be, how shall I say, prevented from being carried out. So that was in the year of our Lord, 1986, when I went through that medical school debacle. At any rate, now you've heard it, and you've had the lab. Congratulations. Hopefully we won't be getting a visit from PETA or any other entity that would cause us from stopping the, how shall I say, the injury to animals. So this lab was performed without injury to animals. It was a piece of white paper and some ink and a little storytelling by the video here. So I hope you enjoyed it. Please inquire with your questions or make comments as you wish as a response to this in our daily episodes together. Thank you very much.